In this video, I'm going to talk about the role of toll-like receptors, and specifically toll-like receptor 6 in the innate immune system. To do so, I'm going to break down this complicated process into 5 steps. Step 1, recognition. So a toll-like receptor, or TLR, sits on the membrane of an immune cell. In our case, I'm going to use a macrophage. The TLR remains inactive until it recognizes a foreign molecule, and in this example, I'll use a bacterial lipoprotein. TLR6 will actually team up with TLR2 to form a dimer, which brings us to step 2, activation. Once the dimer, which is just 2 molecules linked together, is formed, it becomes activated. From here, the dimer can carry out the necessary functions to eliminate the threat. To do so, the 2 TLRs initiate a series of events, or a cascade, that are pretty complicated, but ultimately this third step leads to the activation of transcription factors, which is step 4. In our case, TLR6 activates NF-kappa B, which stands for nuclear factor kappa B. This will encourage DNA to transcribe new RNA, and then this RNA will be translated by ribosomes to make pro-inflammatory cytokines. These are basically small proteins that induce inflammation. The main cytokines produced here are TNF-alpha, IL-1B, and IL-18. TNF-alpha is ready to go, but IL-1B and 1A are inactive, so they have to be activated by an enzyme called caspase 1. This guy basically cuts the 2 IL cytokines in half, and now they're activated and ready to go. This leads us to our last and most important step, attack. So, TNF-alpha and IL-1B and 1A have their own independent functions in the immune system, but the 3 most important functions they have are enhancing immune responses, inducing programmed cell death, or apoptosis, and activating immune cells such as lymphocytes. The lymphocytes can then attack and hopefully destroy the bacteria that activated the TLR6 in the first place.